already heard about breast cancer, so I will skip this and just go away from there. Well, you can have a susceptibility gene, but you can also have something else beyond the susceptibility gene. This is what I meant by saying that we know that there is an enzyme which actually monitors the epigenetic silencing of the genes available in our body, and that unfortunately an epigenetic silencing in those genes which regulate cell kinetics, regulate cell division can also be silenced. So the cancer is actually caused by an epigenetic phenomena, not the actual business of altering in the gene itself. So DNA methyltransferases are equally important. So there are pretty many DNA methyltransferases, but they are seen in many cancers. Can we use them as pharmacogenomics or pharmacogenetics? If pharmacogenetics is the whole population, yes, you can study the whole population like we did for the cytochrome P450 enzyme systems, first for warfarin, then for many others, including drugs like morphine, codeine, etc. You can have a rapid metabolizer or a poor metabolizer or an intermediate metabolizer, and that can give you enough information to say that this is the toxicity to expect from the drug you are choosing, or this is the efficacy or resistance to the treatment which you are planning to use. It's pretty slow. Okay. So this is what I meant by what is tamoxifen metabolism. So you can have an extensive metabolizer or a poor metabolizer or an intermediate metabolizer, and Asians fall into the intermediate metabolizer group, and they have a worse recurrence rate, decreased progression, relapse-free survival, worse progression-free survival in the adjuvant setting. So we know that this information is going to be pretty useful. So this is what I meant. So if you can plot it against the time, and you know what is the genotype which is available, EM over EM or EM over IM, doesn't matter, don't worry about that, what that is. So you can split them into 3 different groups, those who do wonderfully well, those who are not going to do very well. So the algorithm, once again, is each time you have a recurrence of the tumor, go back and again, and consult the laboratory to see, yes, this genetic information changed. Cancer is one disease which doesn't remain exactly the same since its inception till it finally carries away your patient. So it is remarkably dynamic. It is remarkably efficient in getting away from you. So use this algorithm and schedule your biopsies accordingly. So get the material. If you can't schedule a biopsy, it doesn't matter. Now you can isolate your circulating tumor cells. If the ICMR doesn't like circulating tumor cells, write a protocol on circulating DNA. They can't run away from it now. So you have abundance of circulating DNA and you can use it. So can this information also be useful for research and drug discovery? Yes, you can now help in enriching the population which enter the clinical trial. So you have a run-in period in which you are enrolling patients and you do a genetic analysis. The results will come back in probably 3 weeks to 4 weeks, and now you can have an enriched population to say, yes, this is the group which should enter this clinical trial. This is exactly how we discovered ALK and we discovered the Crizalk for it. So exactly the way that a small percentage of patients, not more than 2, actually responded to the treatment, and we were wondering why those patients responded to treatment, and then we discovered. So we first discovered the drug, we discovered the mutation later, and then the enriched population was what was helpful.